Thank you very much for the invitation to provide this overview of some of the exciting developments in multiple myeloma, based in part on abstracts that were presented at the 2024 American Society of Hematology, many of which have now been published in the peer-reviewed literature. My name is Bob Orlowski. I'm the Florence Maud Thomas Cancer Research Professor at MD Anderson, serving as the Deputy Chair and the Vice Chair for Translational Research in Multiple Myeloma. From an overview perspective, I think we can start with the earlier lines and then move to more advanced disease. And it doesn't get too much earlier than smoldering multiple myeloma. I'm sure all of you know that for right now, we don't yet have anything approved for the smoldering setting. And it's definitely an area where we can improve upon, because many of these people, especially if they're at high risk of progression, can develop bone lesions, anemia, renal failure, and yet up to now, the standard of care is still a watchful waiting approach, which of course is quite frustrating for patients as well as for providers. So we did see a study presented at first the International Myeloma Symposium and then at ASH, and now has been published in the New England Journal. Thanos Dimopoulos was the first author, and this was a study looking at smoldering patients who were randomized to receive either watchful waiting, also called active monitoring, or to daratumumab as a single agent, and then monitored moving forward. In terms of progression-free survival, which was the primary endpoint, patients who got daratumumab had a lesser likelihood of progressing to symptomatic disease. You can see from the graph that there's a nice separation of the curves in favor of daratumumab, and that separation really begins to happen very early in the trial and then seems to actually enlarge over time. And although the overall survival data were not yet mature, it did look like there was a trend beginning to favor the daratumumab arm. So based on these data, we're hopeful that we will later this year get an approval from the FDA to have DARA available for smoldering myeloma. Probably this will be for high-risk smoldering, according to the IMWG 2 2020 system, where the 2 means more than 2 gram per deciliter M protein. One of the 20s is more than 20% bone marrow involvement, and the other 20 is an involved to uninvolved free light chain ratio of at least 20. Any 2 of those put you into the high-risk setting, and DARA, again, will hopefully be the first agent approved. We also expect that there will be other combinations available down the road, including anti-CD38 antibodies, either DARA or isatuximab with lenalidomide and dexamethasone. And in terms of the toxicity profile, for those of you that have used daratumumab, there really were not any surprising findings. The group that were randomized to DARA had a little bit more fatigue and some risk of upper respiratory infections, but otherwise it was a very well-tolerated therapy with a relatively low incidence of serious adverse events. Moving from smoldering now to newly diagnosed myeloma. And of course, for symptomatic patients, we still divide people up into those who are transplant eligible, and we'll start with that group, versus folks who are transplant ineligible, who we'll get to in one of the subsequent slides. The first study that really was very interesting came from the German study group looking at these transplant-eligible patients and randomized them to get either one of the current standards of care at that time, which was RVD or lenalidomide with bortezomib and dexamethasone, versus the addition of isatuximab, an anti-CD38 antibody. And this trial looked at both progression-free survival as well as minimal residual disease negativity. This was a large study, and you can see from the graph that the ESA RVD group in the purple color had a better progression-free survival than did the folks who got just the triplet RVD in the red color. And MRD-wise, what was interesting here is that if you were able to achieve MRD negativity, whether it was on RVD or on ESA RVD, you did better than if you did not achieve MRD negativity, which of course is expected. Less cancer is always going to be better than more cancer. But the interesting thing is, of course, that with ESA, there were more patients that achieved MRD negativity with the quadruplet than was the case with the triplet. One of the interesting things now in terms of MRD testing is, are there other approaches that we can use besides just bone marrow evaluations? We can do MRD either with what's called next generation flow cytometry, which is good for about 1 in 10 to the 5th cells, or we can do it based on sequencing with the typically used clonoSEQ assay, which gets you down to about 1 in 10 to the 6th cells that can be detected. But of course, bone marrows are not that easy on patients, and it would be interesting to have an even more convenient way. One approach that we think is coming is to do mass spectrometry on the peripheral blood, which would allow us to do serial assays and be more sensitive than approaches relying on just the immunofixation of the serum. But there was an analysis presented at ASH from the Perseus study, which was DARA RVD versus RVD, where they looked at circulating tumor cells. We know from past studies that patients can have low levels of circulating myeloma cells that can be detected in the peripheral blood. And the interesting thing about that kind of assay is that not only can you detect how much tumor there is, but in some cases, some of the assays allow you to do molecular testing on those cells. So you get an evaluation not just of disease burden, but of the biology of the disease as well. So in the analysis that was presented at ASH, there were a couple of things that were interesting. In the top graph, what you can see is that higher levels of circulating tumor cells were associated with a more rapid progression to the next line of therapy, which is to be expected. And people with very low levels of circulating tumor cells did better. The second analysis that I thought was particularly interesting is that they looked at whether this was an independent prognostic risk factor compared to molecular risk as well as ISS stage. And they did actually confirm that the percentage of circulating tumor cells was an independent risk. So hopefully this is something that will be available moving forward, where we can, first of all, with greater sensitivity, detect how much myeloma is left, and also maybe make some decisions in the future about treatment. For example, people with more circulating tumor cells may need more therapy compared with people that have fewer circulating tumor cells. There also were studies looking at the value of these circulating tumor cells in the setting of getting quadruplet versus triplet therapy, and also high risk versus standard risk. In terms of the risk profile, essentially having low levels of circulating tumor cells was beneficial, whether you had standard risk or high risk, but the difference was particularly important in the molecularly high-risk patients. Moving on to the Cepheus study, which was DARA VRD versus VRD in transplant ineligible patients. This was an attempt to show that quadruplet therapy could be effective and safe in this transplant ineligible population. We know, of course, from previous studies that have been presented that this is true for transplant-eligible patients. Here in Cepheus, again, focusing on transplant-ineligible, newly diagnosed myeloma, really the main exclusion to keep in mind is that most of these patients would not have been in the frail category, but they were randomized to get either the triplet or the quadruplet. And as you probably would expect from the transplant-eligible population, in the ineligible patients, the quadruplet did better in terms of progression-free survival, and minimal residual disease negativity was higher with the quadruplet. This was true at 10 to the minus fifth, at 10 to the minus sixth, and there also was a better rate of sustained MRD negativity. Sustained MRD negativity means that on at least 2 consecutive measurements, patients were MRD negative. And as you would imagine, people who have sustained MRD negativity do better than people who have MRD negativity just at a single time point. The other trial that really, I think, informs what we do for transplant-ineligible patients was the IMROS study, which also looked at VRD as the control arm and compared it to isatuximab VRD. Here, the quadruplet again was superior with a hazard ratio of 0.6, which means a 40% reduction in the risk of progression. Full disclosure, I was one of the authors and investigators on this study, so the data are exciting and hopefully will change what we do in transplant-ineligible patients. And MRD negativity, again, was better with the quadruplet than was the case with the triplet, both in the entire patient population as well as in those with complete response. And the sustained MRD negativity rate was almost double. So this is really what you want to try to achieve in patients, is this MRD negativity that is sustained over time. And the adverse events were pretty much what you would expect with an anti-CD38 antibody. In addition to IMROS, there was an interesting study which is in some ways a complement, and this was called the BENEFIT trial. In this study, the control arm was isatuximab with Lendex, and the experimental arm was ISA-VRD. So it's another test of a quadruplet in transplant-ineligible patients, but the difference here is that one arm had bortezomib and the other arm did not. So it really asks the question, are proteasome inhibitors still important in transplant-ineligible, newly diagnosed patients? This was an almost 270 patient study by the time you got to the randomization, and you can see that the overall response rate was better for the quadruplet, and the very good partial response rate as well as complete response rate was better if you got the 4 drugs. In fact, the CR rate was almost double, which I think is really important and does show you that for transplant ineligible patients, adding that fourth drug in the form of a proteasome inhibitor can be really helpful. And MRD negativity was better at both 10 to the minus fifth as well as 10 to the minus sixth. And if you look at the forest plot there, you can see that the quad was better in almost all of the subgroups, including men and women, younger ineligible patients versus, how shall we say, more mature patients, and in standard risk as well as molecularly high risk patients. For people that have had a transplant after prior therapy, for many years, lenalidomide as a single agent has been the standard of care for maintenance, especially if people are standard risk molecularly. We did see data at ASH that are now published from the Auriga study. And in this trial, patients after transplant who still were MRD positive were randomized to get either len alone or they got Dara plus len in combination and then were monitored for progression-free survival. Pretty large study with several hundred patients that were enrolled. And the Dara-Len arm, which is in the red curve on the right, did show a better progression-free survival compared with just Len alone in the green arm. So again, if you're MRD positive still after transplant, it does suggest that the combination is going to be better than len alone. And we do hope soon to have data from a SWOG study that finished enrollment earlier this year that looked at len versus len-dara as a maintenance, although it took all comers. So you could be either MRD negative after transplant or MRD positive. It may still be a couple of years before we know, but that will really be the last question to ask in the maintenance setting. And I do think that definitely for MRD positive patients, you should consider the combination, and potentially based on the SWOG study, maybe even for MRD negative patients as well. From a side effect perspective, Dara and Len was pretty well tolerated. And of course, MRD negative patients were able to do better than patients who still stayed MRD positive despite the maintenance that they were on. Another interesting aspect is MRD 1 year after stem cell transplant. And one of the studies that looked at this and had been presented at ASH was later published in the Journal of Clinical Oncology. One of the things that they looked at is if you are MRD negative at 1 year after transplant, does that translate into better outcomes? And as you might imagine, the answer is yes, in terms of progression-free survival as well as overall survival. Another question that often comes up in this area is, well, if you have MRD data, what should you do about it, especially if you're MRD positive? So they did an interesting analysis, which is that they looked at the patients who were MRD positive, but then converted to MRD negative, and whether their outcomes were different than the people who stayed MRD negative throughout. And the interesting thing there is that both on PFS as well as OS, it looked like the people who converted to MRD negative from a previous positive did as well as the people who were negative to begin with. And that really gives you the impetus to say, not just is the MRD data important prognostically, but it may also support your treating the positive patients a little bit more aggressively so that they can achieve MRD negativity. Moving now to the relapsed and refractory space, there are a number of study updates that we've been able to see recently. And one was an update of the IKEMA study. With IKEMA, you probably remember that the control arm was carfilzomib with dexamethasone, or KD, and the experimental arm was isatuximab with KD, or IKD. And the early results of that showed a strong benefit for progression-free survival and also a trend towards a better overall survival. Towards the end of last year, we saw publication of the OS analysis, the final overall survival data. And again, progression-free survival maintained a strong advantage for the triplet. But now the overall survival advantage, although there was still a trend that favored the triplet, the data now did not quite reach statistical significance. You can see the hazard ratio of 0.855, which means about a 15%, 15% reduction in risk of progression or death, which didn't quite meet the 0.05 p-value, although the trend was there at 0.18. I still think this is a very valuable regimen with a PFS that's over 3 years. And the fact that the OS now is not as positive is really just an indicator of how successful we are at salvaging patients with the next line of therapy if they do progress. Interestingly, there was a similar analysis published of the Icaria study. This was pomalidomide and dex versus isatuximab pomalidomide and dexamethasone. On average, this was a later line of therapy patient population because they had to be refractory to lenalidomide. And here again, progression-free survival was substantially better for the triplet. And the early data showed an OS advantage, and the final analysis also confirmed an OS advantage with a 0.776 hazard ratio, which is almost a 25% reduction in risk of progression, and about an 8-month difference in the overall survival. So definitely something to consider with isatuximab. And I think we'll also go into upcoming data that we'll see about a subcutaneous administration of ISA. Right now, isatuximab is only approved through an intravenous route, but there was a press announcement, and I think we're hoping to see this at ASCO in just a few weeks, of data from the Heraclea study. And this was a trial of a triplet versus a triplet where everybody got isatuximab with POM and DEX, but one arm got the ISA given intravenously, whereas the other arm got it subcutaneously, which is through what's called an on-body delivery system. So this is a device which is put on the patient's body by a nurse, and then the injection occurs automatically. And after the injection, the patient can simply take this device off and discard it. So it's something that I think all of us are really looking forward to. And this study met its primary endpoint. And hopefully as well, we'll see the data as I mentioned at ASCO. This was again a non-inferiority study, and hopefully will lead to the approval of this on-body delivery system. Moving now to therapies that maybe aren't yet FDA approved, but we hope soon will be. all of us are used to using lenalidomide and pomalidomide in particular. There is a new generation of cereblon binding drugs that are in development. The two main ones for myeloma are iberdomide and mezigdomide. These are drugs that were actually rationally designed to better bind cereblon more tightly. And in preclinical studies, they seem to be better than the older generation like len and pom in terms of their anti-myeloma efficacy as well as their immune modulation. So we have an update that was presented at ASH of a couple of combinations. One was mezigdomide with carfilzomib dex. The other is mezigdomide with bortezomib dex. Both of these, by the way, are now in phase 3 trials. So hopefully you will have access to them. And if you have patients that need novel therapies, I would definitely recommend these. But with either mezi and carfilzomib dex or with mezi and bortezomib dex, you can see that these were patients that had a median of 1 to 3 prior lines of therapy, including a lot of patients with stem cell transplant. Many of them had prior proteasome inhibitors, and almost all of them had prior immunomodulatory drugs. And yet they still had, with these combinations, really nice median progression-free survivals that were at least a year to 18 months potentially, and really, I think, promising data. Also, correlative studies that have been done in some of these trials show that these drugs appear to be able to reverse T-cell exhaustion. So that's exciting because if we use a CAR T, or in particular, if we use a T-cell engager, then oftentimes patients will develop exhausted T cells, which may limit what options you have in the next line of therapy. For example, if you use one T cell engager and the patient progresses and they have exhausted T cells, a second T cell engager against a different target may not work as well. But we have data from, again, preclinical correlative studies showing that iberdomide and mezigdomide may be able to reverse that T cell exhaustion. And so that may ultimately allow us to do one T cell engager. If they progress, move to an IBER or MESI combination, both for clinical efficacy and to overcome T-cell exhaustion. And then if they progress after that, switch back to a T-cell engager against a different target. For example, BCMA T-cell engager, MESI combo, and GPRC5D T-cell engager in that sequence of therapies. So I think those were some of the highlights of ASH and recent publications in myeloma. Some of these will inform your practice now, and hopefully many more will be available for you down the road as we try to get to that cure for myeloma. And thank you very much again for the opportunity to review all of this for you.